Greetings, this is Dr. Richard Huhn, Medical Director at Biothera Pharmaceuticals. We are conducting a Phase 2 clinical trial of our innate immunomodulator Imprime PGG in combination with Pembralizumab for head and neck squamous cell carcinoma that has failed to respond to Pembralizumab alone. Our current active clinical program consists of 3 Phase 2 trials of Imprime PGG in combination with checkpoint inhibitor. The first is for non-small cell lung cancer. The second is for advanced melanoma refractory to checkpoint inhibitor and metastatic triple-negative breast cancer refractory to chemotherapy. And the third is for head and neck squamous cell cancer failing to respond to Pembralizumab. Other clinical projects are in planning. Imprime PGG is a beta-glucan, a branched-chain carbohydrate polymer purified from yeast. The active drug is a complex of Imprime PGG opsonized by naturally occurring serum anti-beta-glucan antibody upon intravenous injection and activated complement. The Imprime PGG complex acts as a pathogen-associated molecular pattern, or PAMP, that activates antigen presentation, repolarizes the tumor microenvironment from immunosuppressive to pro-inflammatory, thus activating tumor cell killing and ultimately supporting adaptive immunity. A patient's endogenous serum anti-beta-glucan antibody level may be a reliable biomarker for predicting the likelihood of a pharmacodynamic response to Imprime PGG. Our current clinical program aims to A. Demonstrate the safety and efficacy of Imprime PGG in combination with Pembralizumab. B. Seek evidence that Imprime PGG can increase the sensitivity of anti-tumor immune responses to Pembralizumab. C. Understand the effect of combining Imprime PGG with Pembralizumab on tumor microenvironmental characteristics using tumor biopsy analyses. And D. Evaluate the relationship between anti-beta-glucan antibody levels, pharmacodynamic effects, and clinical anti-tumor outcomes. We are currently conducting a study of Imprime PGG in combination with Pembralizumab in patients with head and neck squamous cell cancer who either have progressive cancer while on treatment with Pembralizumab or who have failed to have a response during treatment with 4 to 8 cycles of Pembralizumab. Head and neck squamous cell cancer is a very prevalent and lethal cancer. Most tumors arise in the epithelial lining of the oral cavity, oropharynx, larynx, or hypopharynx. Early cases often have minimal or no symptoms, and by the time of diagnosis, many cases have spread to local lymph nodes. Surgical approaches are difficult due to the complex anatomy of the head and neck. Keytruda, Pembralizumab, has been conditionally approved by the U.S. Food and Drug Administration for the treatment of patients with recurrent or metastatic squamous cell cancer of the head and neck who have experienced tumor progression on treatment with standard platinum-based chemotherapy regimens. In the KEYNOTE-040 trial, overall survival was improved to 8.4 months from 7.1 months. Our hypothesis is that Imprime PGG, in combination with Keytruda, will further improve outcomes such as tumor response rates in patients with squamous cell cancer of the head and neck. Our trial will enroll patients who have not had benefit from Pembralizumab monotherapy, either with tumor growth while receiving Pembralizumab or by failing to have a tumor response after 4 to 8 cycles of Pembralizumab. Patients must have serum anti-beta-glucan antibody levels of 20 micrograms per milliliter or greater. Approximately 87 patients will be enrolled, 38 with progressive disease and 49 with stable disease. The main objectives are overall response rate and safety. This is the high-level schematic diagram of the study. The 2 groups, progressive disease and stable disease, will be enrolled independently and will receive Imprime PGG at a dose of 4 milligrams per kilogram weekly and Pembralizumab at a dose of 200 milligrams every 3 weeks. According to initial statistical considerations, clinically significant improvement in outcome will be achieved if 5 or more responses are observed in the progressive disease group or if 17 or more responses are observed in the stable disease group. Thank you for your attention. Please feel free to address questions to me by email or phone call.